they will be retained under control. And from there, you can take that bacteria, put that in the plate, and then you can get, even in a diluted sample, you can still get bacterial colonies. Okay, so that's for the initially two methods which we discussed about to access microorganisms. And the third method we are talking about is the most probable number. So the third method is the most probable number, which is mostly to measure bacteria that will not grow on solid medium. In this case, what we do is we dilute the sample repeatedly and we inoculate several drop tubes for each dilution point, and we count the number of positive tubes in each set. This is actually the industrial method which is used to count the number of, the amount of bacterial load in a sample. And this is a very statistical method. It determines a 95% probability that your population falls within a certain range. Okay, so basically if you are actually this is a commercial method also, where you tend to determine what is your CFC per ml. Okay, for example, the RO water which you are using, or any drinking water you are using. Actually, right now you must have heard in the news that when people went to Kumbh Mela, okay, there was a lot of bacterial load in the water. Didn't you hear that news? That the water is impure. Okay, there's lot of pollutants here. How people calculated the bacterial load? It is calculated based on the MPN number. So what is done in an industrial scale, even for any drinking water standard, is you take a sample, you dilute it at in the media at serial dilution. Okay, and from there you calculate. So if you see in your dilution, first three tubes are positive, you will get a count. You will get some standard count. Is there any problem? I see a lot of people not settling down. Is it a problem?
Actually, ma'am, new students are coming. That's why there is.
Yeah, the class timing is 8:30. You should be there by 8:30, isn't it? Doesn't mean the online class will come anytime you want and you can walk in anytime. Okay. So MPN number is the commercial is a number which is even used to determine in a commercial setup that what is the bacterial load in your water. Mostly it is done with water samples. Or if you have to check like how much your river is polluted. You take that, you inoculate that in a serial, you dilute it serially and you inoculate in, for example, 10 tubes. You also have a standard reference for a bacterial load. Okay, so based on that, you calculate that if my three tubes are positive, then my bacterial load is, for example, 100 CFC per ml. So if you take this, all your drinking water bottles, it will tell you your MPN number is less than 10 CFC per ml. So what the government has done is, it based on this MPN formula, it has given a value for your drinking water, and that is calculated by your MPN number. Okay, it could be less than 10 CFC per ml for some samples. If you go to river water, it could be 50 CFC per ml, depending on what is your water body where you want to see the level of pollutant. For example, right now when this Kumbh Mela has happened in UP, the CFC per ml was very high. It was in millions per ml. So it was known that it was very polluted. Okay. So normally in this cases, you check for like, because when the pollution happens, you have lot of fecal samples, fecal E. coli there. Okay. So normally basically you check for fecal E. coli. If your coliform or your fecal E. coli goes into your media, it means your sample is polluted with your fecal samples because of the human intervention. Okay. So NPN number is a very important number for, and you should always check this number for drinking water. There is a range given by the government which you should be using to check whether this water is at a drinking level or it should not be taken by a human. Okay, so this is the standard NPN number used by all the regulatory agencies. Okay, the another method is your direct microscopy count. Okay, so you can actually count the you can see the bacteria under 100x, isn't it? But you need to count your bacteria, that how much of bacteria I have per ml. So in this case, I have a specific volume of a bacterial sample. I place it in a microscopic slide with a special grade that I will tell you now. And you stain your sample to visualize bacteria, and then you multiply by a factor to obtain concentration. Okay. For example, what I have is, this is known as hemocytometer. Have you seen your hemocytometers in your first semesters? How many have you seen hemocytometers? Have you read about hemocytometer? No. Okay. Then I will explain. See, hemocytometer is a very simple instrument. Okay. Imagine a slide with different squares marked on it. Okay, so you have a slide where you have different squares engraved on it, and you can differentiate, because when you see under the microscope, you cannot differentiate which square is what. Okay, so the way you do it, it will give you a very 0.1 millimeter thickness. So when you put a cover slip, you will get a thickness of 0.1 millimeter, and then what you have is your squares with different number of lines. Okay, so how to make you visualize is, so can you see this slide? So you have a slide, you have a square engraved here. If you see this properly, you have lines of different thickness, isn't it? So you have basically, you will have 25 squares each, and each square will divide into your 4 into 4. Okay. So, and then what you will have is, you will have 3 lines here. Okay, this thick line will, when you see under the microscope, you will have 3 lines, while in this case, you will have is 1 line. Okay, so what you have is, at the end, you have, so what you have, this is a, in this square, at the end, you will have 3 lines together. So it will mark the beginning of a square. If you see, all the square are marked by 3 lines, and inside you have, 4 lines. So you have 4 into 4, 16. And then we will have another 25 squares. Sorry. So you have 25 squares, and then each square has small 16 squares. Okay. So now what you do is, you take your bacterial sample. Okay, and you take only 10 microliters of your bacterial sample, and you put it under this hemocytometer, put it under a cover slip. Now what you will see is, all your bacteria will be there in this your slide. Okay, can you see this bacteria now? You can count this number of bacteria. Isn't it? So you count, for example, I'll count this one square. So I will take, okay, so how many bacteria? 1, 2, 3, 4, 5, 6, 7, 8, 9, 11, 12, 13, there are 14 bacterial cells here in this one square. Okay, so what I do is, 14 bacterial square, I have taken 10 bacterial cells. Okay, so my count will become 140 bacterial, 140 bacterial cells per annum. Okay, but the way you do it, you also multiply by the volume. Okay, so the way it is no, I don't have it here. So what you also have is, you also multiply, because you are adding 0.01 ml, which is 10 microliters, and then you will have 10 microliters, so you calculate the volume also. Okay, so and then, so that volume comes to a constant figure of 10 to the power 4, which is 10,000. This is a constant figure in your hemocytometer. Okay, So when you add 10 microliters of your sample, you get a volume of 10 to the power 4. Okay, so what I get is 14 bacterial cells into 10 to the power 4 bacterial cells per ml. Okay, now the problem could be, let me stop sharing. Did you understand the method? So you have 25 squares with 3 lines, and each square will have each subdivided into 16 squares. And then you count one, for example, you count one square with 3 lines. Okay, now with this method, you will face two problems. One is, if my bacterial sample is not homogeneous, I have some clumps of bacteria. So for example, so how do I solve the problem? I count all the sides of the bacterial cell, of the hemocytometer. So I will take count here, here. If your sample is not homogeneous, I will count on both the sides and the middle one. So I will, for example, now I will get 14 into 5, for example. So I should get 90 bacteria in that sample. So when I do the count, it becomes 90 by 4 into 10 to the power 4. Okay, so that is my bacterial cell. My another problem is my sample can be too concentrated. Okay, then I need to dilute the sample. For example, I can dilute it 100 times. Then I have to multiply it by, if I have to dilute it by 100 times, I still get a count, for example, is 14 bacterial cells. Okay, so now I get it 14 into 10 to the power 4 into 100. So it becomes 14 into 10 to the power 6 bacterial cells per ml. Okay, so what you do is you have to count the bacterial cell. You should know the bacterial cell in each square, but you count multiple squares and then take an average. Instead of just counting one square with 3 lines, I count multiple squares so that I can make an average. So there is no problem there. If my sample, if there is some mix, if my sample is not homogeneous, I can still get an average number. So if I have counted 4 squares, I have 100 bacterial cells, for example, I will do 100 by 4. If I count all 25 squares, 100 by 25. If I count only 2 squares, 100 by 2. Okay, and then what you have is, then you put it 10 to the power 4. That's a constant number when you add 10 microliters. Because your volume is, 10 millimeter into 10 millimeter into 10 millimeter. So you get the constant volume of, with that hemocytometer is 10 to the power 4. That number is constant. So if you have not diluted your sample, if you have 100 bacterial samples, it becomes 100 by suppose 4 squares, 25 into 10 to the power 4 bacterial cells per ml. But if, for example, if my sample is too concentrated, I dilute it 100 times, then it becomes 100 by 4 into 10 to the power 4 into 100. Because that is my dilution factor. I have diluted my sample 100 times and then I have put 10 microliters in my hemocytometer. Okay, and this is known. So I will get now 25 into 10 to the power 6 bacterial cells per ml. Okay, so that's the way you count your bacterial cells on a hemocytometer. Are you clear about this? You will be using a lot of hemocytometer in your practicals or in your research. So you should be clear about the concept. Are you clear? Yes or no? If you are not clear, I can repeat it.
Yes.
Yes or no? Nothing?
Yes.
It's clear.
Okay. But what are the advantages and disadvantages of this method? Okay. Advantages, I don't need to incubate the sample. Okay. I can just take my sample and I put it on the hemocytometer. I do not need to do any work. And I could get this count in even 5 minutes. Okay. So this is the advantage here. Now what is the disadvantage? I cannot distinguish between live and dead bacteria. Okay. Like when you do between a spread plate method, then what will happen is if I do a spread plate, I will get, only the live bacteria will grow, and then I will get the number of viable bacteria per cell. And then, but in this cases, I will not be able to differentiate if the bacteria is live or dead. So this is the disadvantage of this method, that I cannot differentiate between live and dead bacteria. Another problem is sometimes the motile bacteria becomes very difficult to count. Okay, because sometimes they will be moving, it may move from one square to another square, then I will do a double counting there, so sometimes it becomes difficult to count those bacteria. And then another problem is you need a high concentration of a bacteria. For example, if I do it in a like the way I talked about water, a lake in Isar Tirupati, it will not have that much of bacterial load. Okay, so I cannot count using a hemocytometer. I need to have at least 10 million cells per ml to count the bacteria. Okay, and that becomes really difficult sometimes. Okay, so these are the disadvantages of this method. This we discussed. Okay, so previously we were discussing about the direct methods. Okay, so when we were trying to count the number of bacteria, we were discussing about the direct method. Where we discuss about, we can take the bacteria, severely dilute it, and then what I can do is, I can plate it on a plate and I will get my CH colony forming units per ml, which we refer as CHU per ml. Isn't it? So what do you mean by direct method is, you will get the number of bacterial colonies per ml. Those are known as direct method. Again, filtration method, I can get a direct number. I can concentrate my sample on a filter and then plate it on a bacterial plate and I will get the number of bacteria. Okay, even with the NPN number, I will get 10 CHUs per ml. Okay, that number also I can get. Fourth is I can directly get by hemocytometer. I can get the direct count of the bacteria. Okay, so now we are going to talk about the indirect method. What are the indirect methods? Where you will not be able to get a direct count, but still you will be able to access growth. Yeah, I see some hands coming up.
Ma'am, a disadvantage of hemocytometer is that there's not enough cells sometimes, right? So why
Yeah.
can't we first concentrate it and then take a count and then multiply it or divide it, like include it in calculations?
Yeah, you can do that also. There is no problem in doing that. So sometimes people do concentrate in the sample and then you do it. So you can get, for example, if you have 100 ml sample, you get it in 1 ml and concentrate it, like in filtration, and then you can do a direct count. That is also a feasible method to do that.
Thank you, ma'am.
Okay. So remember that it's your method of choice. You can now use any of this method to get the number of. But you should also know, like when you count, it doesn't mean that if I get a count of 100 million cells per ml, doesn't mean I will get on the plate 100 million cells per ml. Because in a hemocytometer, my all bacteria may not be viable, they may not be live, they may be just dead bacteria. Okay, so you should always, when I ask, for example, if I ask for in a mid cell, calculate the number of viable bacteria in a sample. Do not put a hemocytometer method. Because I will not be able to say, isn't it? You can say that I can get the total amount of bacterial in a sample is by hemocytometer. But if I have to go to the viable number, I have to still do a plating and check. Okay, so you should be able to differentiate between different methods. Okay, so what are the indirect methods of counting? Okay, so what are indirect methods? One is your turbidity. Okay, all of us do this method. Knowingly or unknowingly, we measure the bacterial growth using turbidity. Okay, so as the bacteria multiplies in the media, it becomes turbid. All of us have seen that when you inoculate a bacterial colony in the night, in the evening, morning, the media is very turbid, and you will see, okay, my bacterial growth has happened. Okay. So you can use sometimes a spectrophotometer to determine the percentage absorbance, and then you can multiply by a factor to determine concentration. This I will tell you. Okay. So how do you do that? Okay. One is when you inoculate a colony in your LB broth, you will see a turbidity in the morning. my bacterial growth has happened. Okay, but you can also use a spectrophotometer to get some number. Okay, like I have talked about, when you talk about confidence cells, you will get an optical density of 0.4. Okay, when you want to do a, by the time you come in the morning, your optical density is 3. Okay, so actually, if you want, you can plot growth curve based on your OD. So OD of 0.4 or 0.5 will tell you that this is in the exponential phase of the bacterial growth. Okay, so what has happened in this case? So what you do is, you take your sample and then you put it under a spectrophotometer. Okay, and you take a reading. Reading is just at 600 nanometers. You are not doing any colorimetry here. You are just maintaining your, you're checking the density of, you're checking the turbidity of your sample. So that's why you use just 600 nanometers. Based on your 600 nanometer OD, you will get 0.4, 0.81. Based on that OD, you will get sometimes, the density of your bacterial cells. So, as your growth is increasing, your optical density or OD will also keep on increasing. Okay, so you keep on measuring your optical density in a spectrophotometer at 600 nanometer, and you can access the bacterial growth. Okay, but this is still an indirect method of counting. I do not know my CFU per ml. If I ask you, I have an OD of 0.5. It will tell you, okay, my bacterial sample has grown, but I do not know CFU per ml. Then how should I find about how to get CFU per ml now? Can you use design experiment with your, what I have taught you, that how will you get from this OD 0.5, how will you calculate the CFU per ml?
If we have another sample where we know the number of CFU in that given sample, could we standardize with respect to that and then directly compare OD value?
You can do that, but that's more, but in real time you don't want to have a standard, isn't it? Although you're right, I'm not saying you're wrong, but you have chosen a difficult method. There could be an easy method also. It's very easy. You take 100 microliters of a culture of your 0.5 OD, plate it on a plate, you will get number of colonies. You will be able to find out. Isn't it? Did you get it? I have a culture of 0.5 OD. I take 100 microliters, put it on a plate. I get 10 colonies, for example. It means I have got 10 colonies in 0.1 ml. In 1 ml, I will get 1000 colonies. So my count is 1000 CFUs per ml. Does it not look okay to you?
Yes.
This is a simple method, isn't it? I don't want to have a reference to do it. I can just take directly culture and plate it on a media, much more easy. I will not waste my reagents also. What I'm trying to raise stress is, you should always think about, you should be able to connect, and if you understand things properly, you will be able to connect. Okay, although your previous method was not wrong, it was also completely fine. Okay, but again, this is a difficult method. Second is, sometimes a 0.5 culture of, it may be a different. You need to dilute it. You also need to follow. You have to follow the same protocol as with this culture. Okay, so you have to think the best way possible to design an experiment. So that's the way you can do it. You can just take 0.5 OD. If I ask you, okay, what will the bacteria load in a 0.8 OD? You take 100 microliters, plate it on a plate. You should get around, triple the amount of colonies. 0.5, or you should get 2000 or something like that colonies. So your CFU per ml should be 2000. It means, okay, fine. Okay, so this is the way you can convert your indirect method to a direct method also, by just doing something which can give you CFU per ml. Okay, but you have to just for example, if you are making competent cells now, okay, which you will be making in the lab, or if you are doing protein production, people have standardized, okay, if I have a 0.4 OD, my cells have just entered an exponential phase, and it will be good enough to give me a good competent cell. If I take for protein production, I go for a higher OD, I go for a 0.8 OD and then do induction. And in that case, okay, I have enough bacterial cells, my cells are in the exponential phase. But if I see a OD of 3, that it means my culture has gone to stationary phase. I can only use it for doing many things. That's the way where people have standardized OD into your, directly doing for experiment.
Ma'am,
Yeah.
the 600 nanometer wavelength that we use, is there a reason for that standard?
First of all, it's the visible range. You can manage, turbidity. But people can use, 550 also, 650 also. 600 is just the best way you can measure turbidity. It's just to measure your turbidity. Like, it's not like the way you do it for, when you do a colorimetry assay. For there, 405 is 405. But for 600, it can be 550 or it can be 620 or 630. You have to just measure in a visible range. So 600, like you have a visible range for 400 to 800. Okay, so 600 becomes the middle of the visible range. But this word is not a hard fast word. But if you are doing a colorimetry reaction, that's a hard fast word. 405 means 405. 380 means 380. Okay. Anything else? So what are the advantages is you don't need to incubation, like if you are growing, you can monitor your growth in the real time. Okay, so if you inoculate in the morning, you can see, okay, after 2 hours my OD is this, after 4 hours my OD is this, so I can monitor the growth in the real time. Okay, but again my problem with this is it cannot differentiate between live and dead bacteria. For example, if I tell you, okay, my OD is 3, it means my culture has gone to stationary phase and I will have some dead bacteria there. Okay, so actually this is sometimes is somewhat, it is perceived differently. When I inoculate a culture, only live bacteria will grow. Okay, so in that time I can assess the growth where I see only, growth, it means my bacteria are live. But when I check a turbidity at OD of 3, it means I'm checking I'm monitoring the measure, I'm monitoring the bacterial growth of both live and dead, because even that time, the culture has gone to stationary phase, the bacteria are dead, but still it is giving me a turbidity. Okay, they will not go from the media. Okay, so again, when you take turbidity, you cannot measure between live and dead bacteria. As such, your turbidity will not measure your live and dead bacteria. But when you are monitoring the growth from initial, inoculation of your culture, initially only live bacteria will grow. So your turbidity will be from the live bacteria. Okay, but as soon as you increase the OD at very high, then you cannot distinguish between live and dead bacteria. Okay, again it requires a concentration of high concentration, which is 10 to 100 million cells per annum. Okay, are you clear about this method? Yeah.
To check about turbidity, you still have to have the sample to be kept in the incubator to get the bacterial cells growth, right? So how come there would be no incubation required, like incubation time period required?
I don't say, for example, you come in the morning, you inoculate your bacteria, and you want to monitor the growth. So what you do is, you have 100, the way you normally do this experiment is, you take a 100 ml culture, okay, I do not know, because you have not prepared competent cells, that's why you are not, aware of this method is, you take a 100 ml bacterial cell, you take 100 ml of LB12, you inoculate, for example, 100 microliters of your culture. So now what will happen is your culture will start growing. Okay, Now, after an hour, you take 1 ml of your sample and you take an OD at 600. Okay, you start getting an OD of 0.01. How much time it took? 2 seconds for me, or maybe maximum 5 minutes. After 1 hour, for example, after 20 minutes, again I take 1 ml of my sample and I monitor my OD. My OD could be 0.1 now. Okay, so I take 1 ml of the sample after 20 minutes because equalized replication time is 20 minutes. By 20 minutes, if the OD can become 0.1 or 0.2, and that will take 5 minutes. So, what this means is taking turbidity will not, for me to measure turbidity takes only 5 minutes. I need to take out the aliquot of the sample and just take OD. So, it means doing the turbidity experiment doesn't require incubation time. Like the way you do it for getting a bacterial colony. Like if I ask you for number of viable cells, then what you have to do is you will take the bacterial cells, plate it on the media, and you will incubate for 24 hours, and then only you will get CFU per ml. Okay, that means there is incubation time for me to know the growth of the bacteria. But to do a turbidity experiment, I need only 5 minutes. I need to take a sample, put it in the spectrophotometer. I will get a reading of any reading 0.1, 0.2, 0.3, and that will tell me also my bacterial cell is growing. Are you clear? Did I answer your question properly?
Yes.
Okay. Okay. Another way of doing it, generally it is not followed in, but you can measure the metabolic activity of your bacterial sample. Okay. For example, they will produce carbon dioxide or they will produce acid. If they're doing fermentation, then they will produce acid. Okay. Then you start measuring the amount of carbon dioxide, or you start measuring the production of lactic acid. For example, I tell you like the way we have done for biochemical classification. I know that if the bacteria is undergoing some fermentation, it will produce lactic acid. Okay, so I start measuring lactic acid in the sample. Okay, so for example, after 1 hour I take a sample, I measure lactic acid. For another example, after some time I will measure lactic acid. So it means I will measure metabolic products. Okay, generally this is not a very common method to use it, and it's very expensive. I need to have specialized instrument to do it. For example, I need to have an instrument that can measure carbon dioxide, and it will depend from one bacteria to another bacteria. Some not strictly carbon dioxide. It will be very difficult for me to have a generalized mechanism for measuring the metabolic activity. Okay, but nonetheless, this is one of the methods to assess metabolic activity. Another method is your dry weight method. Okay. Okay, let me explain you this. What is dry weight? Okay, normally very simple method is I take my sample, I pellet it down, okay, and I dry the bacterial pellet. We do it. If your bacterial pellet is too much, you will say, oh, my bacteria has grown very nicely. If your bacterial pellet is very less, you will say, okay, my bacteria hasn't grown properly. Okay, a very indirect method to for counting. Okay, so you can just measure the bacterial pellet and have an indirect assessment of the growth of the bacteria. Okay, so normally it's a very rough method, but it will tell you, okay, my bacterial growth has happened or not. Okay, for example, if you pellet your culture at 0.4, 0.8 and 3 OD, at 3 OD, I will still get a good bacterial pellet as compared to 0.8 OD as compared to 0.4. Do all of you agree with me? Isn't it? If I take say 1 ml of your culture, if I pellet it down, my OD of 0.4 will be less, 0.8 will be more, and 3 will be higher. But again, my problem will be, although I will have 3 OD as a higher pellet, okay, but I cannot distinguish again because there is some dead cells, some live cells. So I cannot distinguish between live and dead cells in this case also. I know I have lot of bacteria, but how many are live bacteria or viable bacteria, I will not have that idea. Okay, now you can counter argue me that when you do your plasmid, have you done plasmid isolation? Or have you seen plasmid isolation? How many of you have seen plasmid isolation? I think yes or no?
Yes.
So if I tell you that when you do a plasmid isolation, you put it in the night, isn't it? You put your colony in the night, and then you grow you grow it till morning, and you do it at a OD of 3. So in the morning your OD something optical density is around 3, and then you pellet it down. You will have live and dead cells, bacteria. Then why do you do a mini prep at such a higher concentration, higher bacterial load? Have you thought about it? Why do you still do that? Why do you still take a overnight culture when there is live and dead bacteria both in the sample? Any
Ma'am,
thought about it? Huh?
isn't the OD at an exponential rate, so we wouldn't have much dead bacteria and mostly live bacteria?
See, OD is when it's in the exponential, it is between 0.4 to 1. When you do an overnight culture, you will have OD at the stationary phase.
Okay.
It means you will have live and dead bacteria both present.
Okay.
So why do you still do a plasmid isolation at that OD? It's out of the syllabus question, but I want you to understand because you will be doing this regularly in your practicals or in your research work, which you do not think, but you should be knowing this.
Maybe after the purification we get a good concentration of our plasma.
Yes, that's one of the point. But remember, that's one of the points that you want to have a higher OD, so you get good concentration of the plasma. That's why you want to have a OD of 3. Okay. Another point is you don't want to have that. It doesn't matter for you to live or dead bacteria because as such you are going to lyse the bacteria, isn't it? You put your solution, you put a lysing solution there. If you remember, you put a solution with SDS. So you still lyse the bacteria. Even if it is dead bacteria, your plasmid will still be there. So you can still use it. Okay. So that's why, like for you, when you do a plasmid isolation, it doesn't matter to you whether it's a live or dead bacteria. You pellet everything. Even if it is dead bacteria, you will lyse that bacteria and the plasmid will come out. Okay. But you do it with the overnight culture so that you get good concentration of the plasmid. Okay. So this is one basic difference. But when you want to do a protein production, when you want to see, again DNA is not, that your plasmids are not prone to so much of digestion. They are not prone to so much of breaking. Even if you keep plasmid outside, they will be there stable. Okay, but when you want to do a protein purification, you don't want to do that. You don't want a dead bacteria in your protein purification. Because what will happen is your bacteria will lyse and what will happen is you will start secreting protease. Okay, and that becomes a problem for you. Okay, so when you want to understand this is a very basic difference, it may not be very much related to, you will not get this in your textbook or in your, anything. But this is a very basic research problem which you should understand why when you make protein at an OD of 0.5 or 0.6. And then you just allow it to grow for 3 hours. You do not allow the bacterial OD to increase more than 1 or 2. If your bacterial OD increases more than 1 and 2, it means your culture has gone into stationary phase and you do not want to do that because you will be working with dead bacteria there. But when you want to go for plasmid isolation, your plasmids are very stable. You can go up to OD of 3 and then like this up. Very basic difference which you will not get anywhere. So keep that in mind. Very practical. You will see, okay, so every time you want is, good number of bacteria. You will say, I will give you the OD of 0.1 sorry, I will do a protein production at a OD of 3, induce it at a OD of 3, and I should get more bacteria. But doesn't mean more bacteria is good bacteria. You have to have the judgment process. Okay, for plasmid OD of 3 is fine, but for protein be between 0.6. It should not increase more than this. Okay, you should know when to look for like when to look what stage is important for which experiment. Okay, so dry wetness you can the bacteria is sensitive to create a cell parent, and again the problem is you cannot distinguish between live and dead cell. Okay, so that's the way you can indirectly measure the growth of bacteria. Okay, one second. Okay, so what I have taught you till now is how you can measure the growth of bacteria. Just to give you a brief, overview, what we have talked about is, we have talked about how the bacterial metabolism works. Bacterial metabolism will work if the bacterial metabolism is working. And what we see the growth of the bacteria. And when we talk about growth of the bacteria, the another practical aspect we talk about is how do you measure the growth of the bacteria. Okay, now I'm talking about it. Once you know how to measure the growth of the bacteria, then you have to design ways to control the growth of microorganisms. Okay, because in real life we do not want most of the bacterial cells are sometimes a pathogen. So most of the time we tend to avoid microorganisms in our samples. You tend to avoid microorganisms in your food. You tend to avoid microorganisms in your plate when you do an experiment. You tend to avoid microorganisms. Okay, so what we are going to move to the next chapter is how to control the growth of microorganisms. Okay, how can you, now you know how to access or measure the growth of the bacteria. The next point becomes how do you control the growth of the microorganism and which methods to use. Remember, all methods are good. It depends what is your purpose of doing a control. Do not forget. Do not say, okay, I will choose the best method, like the way we do it for microscopy. Sometimes the best method may not be the method of choice because you may not need it. Okay, so what we are going to talk about is how to, control the growth of microorganisms. Okay, so control of microbial growth is effective in two ways. Either you kill microorganisms, you just kill them, or you inhibit the growth of microorganisms. You make the growth so slow so that they are not able to grow in that area. Okay, so you normally control by killing the microorganisms and by inhibiting the growth of microorganisms. We will be talking about these two things. Killing means cidal. Okay, it could be bactericidal, it could be microcidal. Inhibiting the growth is static. It's a bacteriostatic reagent. It means it's inhibiting the growth of the microorganism. Okay, it involves both physical and chemical agents. Okay, I will just explain to you about these terms. These are very important terms. Okay, do not confuse this. This may look very confusing and it may look same to all of you. But again, industrially, infection biology, this means a lot. Especially when the pandemic happened, these things meant a lot. Okay, what is sterilization? When I say that I have performed the sterilization, it means I have removed all forms of microbial life, including endospores. Okay, it means I do not have microbial growth in my samples. Even if I keep it for months, I will still not have growth because I also avoid endospores there. Now, what is commercial sterilization? Commercial sterilization is in terms of like when you get any food product. Okay, for example, you are having now, you will all be going to US abroad. Okay, you will get lot of tainted food. Or you have lot of you take lot of bottled juices. You take fruity, you take, this, all these bottled juices, you take, paper boat juices. They are very prone to growth. If you take a juice, it is very prone to growth. Okay, but how do the industry define that there is no growth? Because these juices, they are very prone to the growth of Clostridium botulinum. Okay, it's a very nasty organism. Clostridium botulinum is very nasty because it can produce a toxin. So if you even kill Clostridium botulinum, if the toxin is still present, it will lead to the death of the consumer. Even like a small amount of toxin is very lethal to the person consuming it. So the way commercial sterilization work is, what you get in your juices or in your canned food is, it was sterilized till the time there is no growth of Clostridium botulinum. That's a standard organism. They will not work on Klebsiella E. coli. They will work on Clostridium botulinum because that's a standard organism that will tell you, okay, if I do not have Clostridium botulinum, the food is really safe. Okay, so that's the way you describe your commercial sterilization. In your labs, in your hospital, you will not worry about Clostridium botulinum. In your hospitals or labs, you will think, okay, there should not be no microbial life, but there should be you should kill endospores. Okay, so there's a big difference between sterilization and a commercial sterilization. Okay, so you give a sufficient heat treatment to kill endospores of Clostridium botulinum in your canned food. So in this case, what will happen is you will have more resistant endospores of thermophilic bacteria will survive, but they will not germinate and grow under normal storage conditions. Okay, only you will get resistant endospores of thermophilic bacteria, but they will not grow under normal temperatures, so they are not worried about it. They are worried mostly about this organism, which is Clostridium botulinum, because you get toxin from this organism. So if you have endospores, then it will lead to more problems. What is disinfection is, you disrupt vegetative pathogens. Okay, it means you do not kill the endospores, you only disrupt the multiplying forms of the growth of the microorganisms. So what you do is destruction of the vegetative pathogens here, and then you can make use of physical or chemical methods. What is antisepsis? Is destructive vegetative pathogens on living tissue. Okay, if I like, for example, I apply an antiseptic solution to my hands, it means I'm basically doing an antisepsis. If anything I do it on a living tissue, I do antisepsis. And if I don't do it if it is on anything else, like if I want to disinfect my table with ethanol, I will not say antisepsis. I will say I have disinfected the table, but when I put it on my hands, I do an antisepsis. Okay. So that's the difference between disinfection and antisepsis. What is degerming? Degerming is removal of microbes from a limited area such as skin around the injection site. Okay, remember when normally you are given a swab to around the injection site, it means you are just degerming a very limited area. Okay, just like the way you have an alcohol soap swab. What is sanitization is just to improve just to lower your microbial count on eating and drinking utensils to save public health levels. Okay, for example, you put your utensils in a dishwasher and you give it high temperature, then it means you will inhibit your microbial growth, or you put it in a bleach or something like that. Okay, just to explain that in a bit more. Okay, this looks very similar, but the problem is that these are interconnected, because the way you define it, these are medically important terms. Okay, if I say that I have done an antiseptic, okay, if you go to a doctor, oh, I had a burn in my hand and I applied a degerming one. Okay, doctor will laugh at you, because degerming you do it with a non-living tissue. You cannot say, okay, you never say, no, I put a degerming solution in my hand. You always say you have put an antiseptic solution when you get burns. Okay, so antiseptic means, because when I say antiseptic, it means I'm putting solution to my living tissue. Okay. Degerming is I remove the germ from a particular area. Okay, because when the doctor is putting an injection, it's actually punching a hole, and the microorganism should not enter through that hole to my bloodstream. So you normally determine area. You just remove microorganisms from a very small area. Okay, because what the nurse or a doctor will do is going to punch a hole. Okay, or when you are given a catheter, they normally put alcohol so that nothing goes inside our bloodstream. So you just detach a small area. Okay, but when you do an antisepsis, you try to put it on a larger area. Like when you might, for example, if I get a handgun, I do not do a degerming there. I do it because I'm soaking my hands in Dettol. Okay, so that is very important. I'm putting my hands in Dettol, so I'm putting in enhancement in antiseptic solution. Okay, but when you put the same Dettol on your table, it becomes a disinfection. Isn't it? So it matters, but these are very, medically important terms. If I say, okay, I disinfected my table, it means doctor will understand, okay, you have done disinfection, it means you have cleaned the operation table. And another term is what was sanitization. Okay, sanitization is done is normally you just try to, sanitize means you just normally try to reduce the count. I have like for example, what is done on hospital floor. It's not that you do not remove microorganisms by, mopping the hospital floor. You just reduce the number of counts, isn't it? Although people may claim it that you will remove 99% of the microorganisms, but still you will still have microorganisms. You are just bringing the microorganism load down. Okay, that's what is known as sanitization. When you say that you have sanitized the area, it means I have lowered down the microbial count. But when I say okay, I have disinfected the area, it means there is no microorganism. So when you talk about I have disinfected my operation instrument. You always say that you have sanitized the hospital floor, but you have disinfected your, surgery instruments. Because there the microbial count should be 0. You don't sanitize your surgery instruments. You disinfect them. And you sanitize the hospital floor to lower the microbial count. So that's the difference. Are you clear about this? Any doubt? Okay then.